So looking at mitosis. So based on like the research and findings, something you guys did in the book, if you're going to explain like mitosis to me, like what would you say? What is it? What do you do? What's going on? Yeah, cell division, right? And so again, we're looking at cells dividing. Okay, and it's not quite cell division. I'll explain that in a little bit. But when you think of mitosis, you think about cells dividing. What's the whole? When you kind of think about it, there's an overall goal, right? So I'm going to kind of be bouncing back and forth a little bit from here to over here. I would also like you guys right now, if you guys have them in front of you, grab a book. I don't care if you're sharing between you and somebody else, but I want you to grab a book and I want you to open up that book, get to page. Let's go to 156 is going to kind of be the page that we're going to be looking at. Mainly today. I apologize for this. Okay, so we're going to think about cells division. Okay, now the first question we need to ask is, well, what's the overall goal? Why do they need to divide? Say it again. I love that answer. That is an awesome answer. Okay, because what have we always talked about before? Cells have got to divide because they're going to get too big. And so the surface area to volume ratio, right, gets too large. God, that's an awesome answer. Right? Okay. So one, right, we're looking at a high surface area to volume. I don't have to even write down surface ratio. There's just a high. So it's got to divide. Efficiency. Good. Okay, now let's get maybe more to no, let's keep going. Okay, why? Why else? Why else the cells need to divide? Okay, they get too big. Okay, that's one of them. What else? Nope. Huh? Nope. Why do cells have to divide? Think about you, right? Think about how you would be like if your cells didn't divide. So think about why you need cells to divide. What do you got?
To repair stuff.
Okay, good, to repair. Now, what would need to be repaired?
Muscles.
Okay, and muscles are made out of? Which tissues are made out of? Cells. So when you're actually saying that to repair things, it's because what needs to be repaired? The cells actually need to be repaired, right? Yeah. So again, we look at it. Well, why? Why? Because maybe there's some damaged cells, right? Okay, yeah. So basically, they need to simply become repaired. We talk about skin cells quite a bit, right? You get a cut, right? Again, what's got to happen, right? Skin's eventually going to grow over, right? Well, your cells got to divide in that case, right? So anytime you get a cut, nick, scratch, little, booby, whatever the case is going on, right? You're going to have to, again, those cells are going to have to repair. Well, then they're going to have to make more, okay? Would be another reason. Come on, think of one more for sure. We start out in mom's womb, right? Mother's womb, as how many? One. Eventually that one turns to? Two turns into? What is happening to you as an individual? You're growing. Very good. Okay. So now we also kind of start looking at, well, the whole, growth and development, okay, all comes from the point, right, of being able to or have our cells divide. Okay. And the whole point here is that when we start looking at cell division, specifically mitosis, is that we're taking one cell, right, and it is splitting into two. And what do we know about those two brand new cells? Same DNA. We're talking almost the exact same everything that you're going to find inside of it, right? Because again, if we have different DNA, we're going to be all screwed up, right? So we want to ensure that's happening. Okay, so overall goal, what we're looking at right here. Okay, the other part that I think we need to understand, and why I had you guys go through the first little section on there, is it dealt with chromosomes. And I talked a little bit about chromosomes, okay? So remember, in our body, how many chromosomes do we have in our what we call body cells, like heart cells, liver cells, blood cells, that which makes up the body? How many total do we have in our cells? Good. 46 total. Okay, yeah, so we got 46 total. Okay, now remember that you get half from your mom, you get half from your dad, okay? So even though we have 46, what we really do is we break this down into pairs, right? And so, for example, in our body, right, well, if I have 46 total and we break that into pairs, how many pairs would there be? There'd be 23 pairs, right? Yeah, so we actually have 23 pairs of chromosomes because half comes from mom, the other half comes from dad. 23 will come from mom. 23 will come from dad. 23 are found in the egg. 23 are found in sperm. They come together. Basically, things fuse together, and now you get 46 total. So when you look at it, okay, which is we don't have 46 separate chromosomes in our body. We actually have 23 pairs, right? And those pairs code for the exact same thing. Does that make sense to you? So you're going to get like chromosome number 1 from mom. You're going to get chromosome number 1 from dad. Guess what? They code for the exact same thing. Okay, maple syrup urine disorder is one of those on chromosome number 1. All right, I'll keep bringing that up. Okay, you get chromosome 2 from mom, you get chromosome 2 for dad. All right, they're going to code for the exact same thing. So you're going to get all 23 chromosomes from mom, all 23 chromosomes from dad in a perfect world. Okay, all right, so that's what we basically have is we have these total of 46 chromosomes. So when we kind of envision, the DNA inside of our body, okay, we envision this in a couple of different ways. Remember that if I draw out a cell, a normal cell that you guys have looked at, like your cheek cell, for example, right? We've got what on the outside? Cell membrane, right? We've got what right in here? Everybody agree with him? What did he say? He said cytoplasm, right? And then what do we have right here? Got the nucleus, right? And what does the nucleus have in its Sarge? What does the nucleus have in its Sarge? No! What is found in the nucleus? DNA. Thank you for whoever helped her. Okay, we have DNA. Okay, so in this case, again, when we look at it, it's just a dark, dense mass, right? Okay, why? Because all the DNA is just what? Just all strung out, right? Okay, it's all in these strings. They make up this little ball of yarn, right, that makes up the nucleus. Okay, so inside of here, we have 46 chromosomes that are strung out. 23 from mom, 23 come from dad. Does that make sense to you guys? Yeah, Sarge, we good now? Okay, yeah. So as we go throughout today, right, the one thing we're going to do is I'm going to kind of draw the different chromosomes. And I'm going to represent blue with the male. I'm going to represent green with the female. We're going to keep it at that. I was trying to find my red marker today, and I couldn't find it. We're going to go with that. Okay, now, the other part that we're looking at within mitosis, we're actually going to kind of overlook like the cell cycle. We're going to look at the overall cell, what occurs in the life cycle of a cell itself, okay? And so you can see it up here, but I've also kind of, split this up into a nice little, linear, kind of more of an outline little form, and that we can break down the cell cycle or the life cycle of a cell into 3 basic processes, okay? And we've got interphase, we've got mitosis, and then we have what they call cytokinesis, okay? So every cell, barring there's no, death, damage, stuff like that, is going to go through these 3 steps, which, as you can see, include other steps themselves, right? Okay. So, for example, if we're going to talk interphase, so beginning of a cell, and as we go through and talk about interphase, right? Interphase up here would basically be starting right at this point right there. Okay. And we're going to envision a cell, right, that is going to have 46 total chromosomes in it. Okay. Does that make sense to you? We're all good there, right? 46, 23 come from mom, 23 come from dad, right? Okay, now, in the case of interphase, right, interphase basically is the beginning step. It's no different than you guys. When you take a look at the beginning of your life, what are you spending most of your energy doing? Growing and developing, right? Okay, growing and developing. It's exactly what's happening here, all right? So what we can do is we can split up interphase into actually 3 phases. All right, G1 stands for just growth. G2 also stands for growth. Okay, pretty easy. Remember G, remember growth. Growth, G. Okay, G1, G2. But there's another process or step that is separating those, right? And can anybody see, we call it the S phase up here, but can anybody tell me what that phase is called, right, based on looking at your notes? Yeah, what do we call it again? Well, I'd kind of like somebody else to answer, but they're all relying on you right now, which is why when I asked Sarge, he's like, well, I have no idea what I mean to ask the question. You just keep answering for them, which is awesome, but they eventually got to start thinking for themselves. Sarah, what do you got for me? What's it? So you got your notes up? Do you have your notes up?
Yeah.
You do. Okay, so if we look, interphase, right? Here's G1. There's G2. What do we say we have in the middle? Got the S phase, right? Also known as DNA replication, and I have it basically, I'll just get rid of this right here. And I'm going to move this up. What's it called? Synthesis phase. Okay, Sarah, can you tell me what synthesis means? Do you know what it means to synthesize? Now, we've talked a lot about this like the first semester. We talk about protein synthesis, for example. All right, if you're synthesizing something, what are you doing?
Some kind of process, I guess.
Yeah, it is a process. You're right. Okay, I'll give you that. It's a process. No, you're fine. You're good. You're good. It is a process. Okay, and I asked this question a lot to this class, so I'm kind of putting you on the spot. Can anybody help Sarah out a little bit as far as what does synthesis mean? If you are in the process of protein synthesis, what is happening? Yeah, making proteins. So what does synthesize mean? To make something. Very good. So in this case, right, the big question is, if we're synthesizing, what are we making? Making what? Yeah, making more DNA, right? Okay, and how do you know that? One, because it tells you right here, DNA replication. And what do we know the process of DNA replication is? Copying, making more of it, right? Okay, yeah. So when we take a look at that synthesis phase, here's where now replication is happening. And you guys already know about replication in the overall process and how we build, right, or copy off DNA. Okay, but now something happens to the amount of DNA that we have inside the cells right here. As I told you guys before, we generally start out with how many? 46, right? 23 pairs. Now, if there is DNA replication happening and your DNA is copying, at that point in time, how many chromosomes do you have in the cell or in the nucleus? If you are copying or replicating DNA and you originally have 46 in it, what does that number now become? Yes, what is 46 plus 46? 92. You're doubling it, right? You're doubling the amount. I just spit, that was disgusting. You're doubling the amount, right, of the amount of DNA. So you don't have 46 anymore. Now you have 92, right? Okay, and so rather than having, again, these single strands, now we've got multiple strands, right? Now we have basically a pair of dad's chromosome number 1. Now we have a pair of mom's chromosome 1, and so on down the line. Okay, we start to see that during that synthesizing or that replication process. And then eventually it's going to jump back into growth. And more along the lines of the growth or G2 phase, it's all about getting ready, right, for cell division or getting ready for this process of mitosis, we'll call it. Okay, all right. Now, jump into mitosis. Okay, now here's the thing about what Owen said earlier that is not correct. I asked a question of, well, if you take mitosis, right, what is, what is it? Okay, what's the overall goal of mitosis, so on down the line? And he said cell division, which is not completely correct. Okay, if you guys remember, hopefully you remember, maybe not all remember. We'll see. I don't know if you gained anything from the worksheets or not. But if we take a look at what mitosis is, it is actually where the nucleus splits. I don't like the word splits. We're going to say the nucleus divides. Okay? It's not the process of the cell dividing. It's more about the contents of what is inside the nucleus and the nucleus itself dividing. Okay? And then the next question is, well, where does the cell actually divide? And that happens down here in this process called cytokinesis. Right? What's up? I like glasses.
We're just
both of you guys.
academic pictures.
Oh, we got another one.
Yeah. Who's the other one?
Brooke. Oh, it's Brooke.
Yeah.
Just Brooke.
You taking pictures now?
Yeah. Just
Everybody
keep
smile.
talking.
Oh, I got to keep talking?
Yeah. Just
Okay.
don't look at
I
the
can't.
camera.
I'm kind of mesmerized by the pink and the purple glasses right now.
What
So
about the blue?
I didn't see them, but she didn't come in. Those are nice as well. Okay. All right. So
Thanks.
cytokinesis, right? Cyto referring to what? Cytoplasm. Okay. Yeah. So if you got cytoplasm, right? Kinesis is probably referring to what? What is happening to the cytoplasm, or what would be happening to cytoplasm and cytokinesis? Lisa. Your kids will, huh? Yeah, it gives a divide. Very good. Yeah, so the cytoplasm divides, right? Okay, so when we look at what's happening in cytokinesis, now here is where the actual cell splits or divides. Okay? So we're going to talk about that process from basically where we look at one cell and how that simply divides into two cells, right? By the process of mitosis and also the process of cytokinesis, right? And not really a huge process there. Okay. Now, here's the other part that we need to understand. I have written over here, 1, 2, 3, I've written over here 6 terms. Okay, 6 terms that are going to come up here in the next probably 10 to 15 minutes, right, that we are going to go through, right, that you should be able to acknowledge or somehow indicate, see, draw, find in a picture. I don't care right, in order to acknowledge what step, right, these cells are in, right, in the process of mitosis or cytokinesis. Okay, let's talk about the first one right away. So remember, we're talking chromosomes. Okay, and in the process of chromosomes, I've already told you guys that generally we're going to have like one copy of male, one copy of female, right? For a total of 46. So let's say, for example, that a cell, not the human body, we're talking about just a hypothetical organism, that they have only 4 chromosomes that make up their cells. Okay? So there, how many pairs would there be? 2 pairs. Okay, you guys with me on that, right? So 2 pairs, we'd be looking at, well, we've got one that comes from mom, right? Or a pair that comes from mom. We've also got a pair that comes from dad, right? Be kind of looking like something like this. Because all it is a string of DNA, right? Okay, and so if this is my chromosome number 1 right here, right? These chromosomes are going to code for the same thing, right? If this is chromosome number 2, they're going to code for the same thing as well, right? Okay, what happened during replication? You guys already told me. When replication occurs, what happens to the DNA, Reese?
It's being copied.
It's being copied. Very good. So what we end up getting is we don't just have one strand of this DNA. Now we end up getting what? Right? Made a copy. Did you guys see that? Yes. No. Okay. So it's the same. Okay, because they're both male that's copied each other, right? So these 2 little strands right here are the exact same copies, unless what occurs? Mutation occurs, right? These strands right here in the female are exactly the same, replicated during S phase, right? Again, down here the same, down here the same. But something else happens though, where they start to join together, okay? And they're going to be joined together, right, by a structure or at an area called the centromere. Okay, so I'm going to enlarge this a little bit over here. All right, so if there's a chromosome, there's a chromosome. There's this little area right here that it's called the centromere. Okay, and that's where they're going to be joined at, that general region. Okay, now, so if this is the centromere region right here, okay, these are what you call sister chromatids. Okay, why are they called sisters? Lisa and Mary, why would they be called sisters? Yeah, and they're pretty much the same, right? Yeah, I mean, they are the same, okay, in this case. So sister chromatids, right? And they are the same. Okay, so here's sister chromatid. There's a centromere. Got that taken care of, okay? Case of nuclear membrane, I shouldn't have to talk much about nuclear membrane. I will bring up spindle fibers in the centrosome here in a little bit. I will bring up cleavage furrow, and I will bring up cell plate here in a little bit as well, okay? But again, when we are going through, you want to be looking at the nucleus. We want to be looking at where spindle fibers are. We want to be looking at what's happening to the nuclear membrane, right? And that's going to be really good indicators as far as, okay, what stage or what process they're in. All right, so let's jump down here a little bit. We're talking mitosis. Mitosis is broken up into 4 phases. Okay, we've got prophase, metaphase, anaphase, and telophase. PMAT, basically just for short. Okay, now you guys look in the book on page 156. Right, those 4 steps is virtually all that you need to know. All right, but we're going to take you through again step by step some of these things and where they're at and where they're in relationship to each other and are they there and so on, which is going to help you to indicate whether it's in prophase, metaphase, anaphase, and telophase, and so on down the line. So if we look at prophase, okay, I'm going to start right here. What do we see is happening, or what do we see, right, is going on with the nuclear membrane? First question is the nuclear membrane there? Is the nucleus there? In that picture on 156, where it says prophase. Yes, it is there. Okay? Now, but something else is happening to the nucleus in prophase. It's still there, but the nucleus starts to, disintegrate. Very good. Which means that it's starting to break down, right? It's not going to be there. Because in order for the DNA to split, what's going to happen to the nucleus? It can't be there, right? It can't. So it's going to disintegrate. Okay? What do we notice about the chromosomes? What do we notice about, just the DNA inside? Is it a ball of yarn and mass that we've seen with like the cheek cells that you guys have looked at? Or can we kind of start to identify individual chromosomes in that picture? You guys tell me. In prophase, what do you see? Do you see just mass, or do you actually see individual chromosomes? Individual chromosomes, right? Yeah. So we start to see is that the chromosomes start to condense, they call it, and start to form. And we say form because these are what we think of when we think of chromosomes. Okay? Not the stringing around the stars of nucleus. It's like whenever people think of chromosomes, it's the X for whatever reason. Okay? So the chromosomes are going to condense, right? And then last one we're going to talk about are these spindle fibers or these centrosomes. Okay? And do you see that they are labeled in prophase? Are they labeled in prophase? Yes, they are, right? They're there. Okay? And so again, you've got this centrosome, you've got these spindle fibers, right? I'm just going to put down spindle fibers up here because we start to see the appearance of things that are going to be useful, right, in the case of cell division. Okay? So there's prophase. How about metaphase? What do we see happening in metaphase? Okay, let's go here. Nuclear membrane. Gone. None. Not even, you don't see it at all. Okay? But probably the key defining, I can't see a picture, that's not what I'm looking for. Structures in this case would be your chromosomes. Because what do we see happening to the chromosomes in metaphase? Yeah, they line up in the middle. So key word there was what, Sergeant? I don't mind that one either. I'm thinking middle. Okay? Because what's the phase called? Phase is called metaphase, right? Metaphase starts with an M. Okay? Chromosomes lined up in the middle, right? Starts with an M. All right? So I'm going to basically put down, all right, chromosomes line up in the middle. Okay? Now, what do we see happening to the spindle fibers? What's a little bit different or unique about them? Where do they go? Yeah, they go to poles, right? They go to the opposite ends, right? Opposite ends of the cell. And hopefully you guys can see that, right? That they're basically sitting there and they're waiting, right, to simply split the chromosomes up, all right? So the metaphase, again, chromosomes line up in the middle. Really the only thing that you need to know. The spindle fibers, yes, go to the opposite poles. How about anaphase, right? What do you see happening in anaphase? Yeah, I mean, we probably should have been calling these sister chromatids all along, but let's just call them chromosomes anyway. But the chromatids separate. What is separating them? Can anybody see? What's it look like?
Looks like fibers.
Yeah, it looks like little fibers, right? Hopefully you guys can read. You can see again, in that picture that you've got these little fibers that are probably pulling them apart, right? Okay, so in the case what's occurring, right, is when you think of basically these spindle fibers are kind of like fishing line, right? Basically, like if somebody's going to throw out a line, fish attaches to it, right? What do you do with the rod? You end up reeling it back in, right? Okay, and that's exactly what you're looking at here, right? In a very, very basic way, right? Is that when you take a look, right, those spindle fibers are attaching, right, to these sister chromatids, right? And they're going to shorten up. And what are they going to do? They're going to split those apart, right? Okay, and so as they split them apart, notice that they're basically pulling, right, half the chromosomes to one side and the other half to the other side, right, ensuring that as cells divide, they're going to get the exact same copy, right? You guys with me on that? Okay, now, if we look at telophase, okay, what's happening in telophase? Any more from you guys? Okay, shouldn't have to sit up here. I mean, we could very easily do worksheets and stuff out of the book, but figured we'd talk about it a little bit. What's happening in telophase? What's going on? Yeah, you get new nucleus form, right? Okay, very good. Nucleus reappears. Okay, what else? I mean, chromosomes are already at opposite ends of the poles, right? That's what happens over here in anaphase, right? What else do you see? Yeah. Okay, you got that going. Who else was saying something? Yeah, okay, so here's where we've got. So now here's where we're starting to look at a little bit of a difference, okay? So if we're talking about an animal cell, right, the one thing that's going to start happening to those cells is that you're going to start to see a little bit of a division. So it's like they're basically taking their fingers and they're going to pinch a balloon in half, right? Well, what do you get? You get one kind of ball on this side, right? And you get one ball on this side over here, okay? Well, they basically have given that little area, right, a term that's called what? Yeah, not to be perverted, but what does it look like? It looks like cleavage. Okay, exactly, right? Yeah, so it's called a cleavage furrow, okay? Because again, the whole cleavage is because you've got that little indentation right there, okay? So as telophase continues, that cleavage is going to get deeper and deeper and deeper and more predominant, right? Until all of a sudden it pinches it off completely and you've got two brand new cells. But where it pinches is a completely different process that we call cytokinesis, okay? But there is a difference because Cole, you told me that you can also see a cell plate, right? Yeah. Because remember, if we're talking about plant versus animals, right, there's a little bit of a difference. Because what is a plant cell surrounded by? Cell wall. So do you think it would be very easy to take and pinch that cell wall like we can an animal cell? Probably not. Yeah, so here's the difference. Okay, in a plant cell, right, it's going to look something like this. You're going to find your chromosomes over here in your nucleus, chromosomes in your nucleus. There's going to be what they call a cell plate that is going to start to form. Okay, and the more and more, the further this gets into telophase, the more complete that cell plate gets until all of a sudden cytokinesis is reached and it's a brand new cell wall. So the differences that we're seeing here in the plant is that there is a cell plate. In the animal, we have cleavage. Okay. Two brand new cells. How many chromosomes do we find on each of those brand new cells? Each cell is 46. So as you can see what happened, in numbers of chromosomes, we go from 46, duplicated or replicated to 92. By the process of mitosis and cytokinesis, we get two brand new cells with 46. What would happen if replication didn't occur? It would be half. So Claire, instead of ending up with 46, how many would we end up with at the end result? If replication didn't happen, we end up with 23. Okay? How about when those cells divide? If there's no replication, what would they end up with? Like 11 and a half, maybe a 12, maybe, I don't know how it would work because it doesn't work that way, right? And then how would they? Well, they're going to break up in odd numbers as well. So as you can see, replication is very, very important because, again, it's going to help to ensure that we have the exact same amount of chromosomes, right, and number, right, in each of those cells. Again, we know that there's mutations. We know that there's things that can happen that can ultimately lead to, that to not occur, to be different, okay? Questions from you guys. So, overall goal, should know why. Okay? You should be able to go through and explain each of these steps, right? But by using terms like sister chromatids, centromeres, nuclear membrane, spindle fibers, right? Cleavage furrow, cell plate, things like that, right? We're not asking you to get too far in depth with this by any means, right? Acknowledge a picture, see the picture, right? And be able to acknowledge what it is, right? And then again, be able to acknowledge the whole cell division and number of chromosomes that will form, all right? Now, we're also going to come back. We're going to talk meiosis on Thursday. Meiosis is a little bit different process. Okay, here we're talking about body cells, cells that make up the body. In meiosis, what kind of cells are we trying to produce? Those that are going to reproduce. They're going to make a baby, right? Okay, and there's a difference there because we don't want the same DNA in reproduction. We don't want to have 46 in each of those cells. And we'll explain that process because what we want genetic diversity in meiosis. We want to have a cell with half as many chromosomes because it's going to pair up with another. They're going to get fused together to ensure to make that 46. And so we'll talk about that a little bit more as time goes on, but there is a big, big difference there. Okay, so how much did I completely screw you guys up today? You guys are fine? You good? Okay, all right. So here's what I got planned for you guys today. I was going to just give it to you tomorrow, but we're going to give it to you guys today. There is going to be an assignment. I don't care. Work on it with part of whatever the case is. I'll give you time to work on it tomorrow, but I really want to. Probably with about 10 minutes left of class. Remember, it's a shortened period. Tomorrow we got 30 minutes. We've got 10 minutes left of class. I want to go through how to grade these in class and be done with it. I did have a couple individuals that were completely done with it before they walked out of here today, which is whatever. It is what it is. But I'll probably end up giving you about 15 minutes tomorrow just to finish up what you need. Okay? So you've got books that you can use. Page 156 probably help you out. Just pass it back, please. You've got notes now that you can use. 6. That's all that color needs it one.
Okay.
Go.
Are you just gonna leave it open for Friday on Wednesday?
Pass those back to us.
There you go.